as our MindCORE speaker, Dwight Kravitz, who is the Associate Professor of Cognitive Neuroscience at George Washington University. Dwight and I actually have known him for quite some time. He was an undergrad at Carnegie Mellon. I didn't know him then, but when he graduated, and he was or close to graduating, he was applied to graduate school. And he actually applied to Penn. This is my first year as an assistant professor, and I got to interview him. I remember after the interview thinking, wow, this guy's really smart. He's really sharp. I hope he comes to Penn to work with me. Well, he was really smart. He actually chose to stay at Carnegie Mellon and work with Marlene Berman and did his PhD there. Did some really nice work on position invariance among other topics. And then he went down and did his postdoc at the National Institute of Mental Health, working with Chris Baker, where he did some just really beautiful studies using fMRI to look at how scenes are represented in the brain. Studies that I think are sort of exemplary in how you actually use fMRI to ask a question about representations, and those studies inspired me for quite some time. And also two sort of landmark review papers, one of the ventral stream and one of the dorsal stream, that have really been seminal in the field of our understanding of how vision is organized. Since 2013, he's been at George Washington University. He's been working on a variety of topics, now expanding beyond just visual perception into working memory, among other topics. But he continues to be someone who I think is someone who thinks very deeply, not just about his specific topics, but how we use the tools of cognitive neuroscience to ask questions. And so I think his research and thought is really fundamental. And so we're going to hear a little bit about that today. Title of his talk, as you can see, neural anatomy, plasticity, and behavior. And are we having any kind of, there's something on them? Yeah, okay. And as soon as we click this, got it. Thank you, Heather. I think it's all working both in the virtual world and in the real world. So Dwight, take it away.
Thanks, Russell, for that really great introduction, and to everyone for the invitation. And it's really my pleasure to talk to you today. Let me know if there are any problems, hearing me or whatever it is. You can shove stuff into the chat or just break in. I'll also say, Russell, your introduction is really perfect, because a lot of what I'm going to talk about today are some of the exact things that you just introduced. What I'm going to try to do is put it into a broader context, specifically looking at exactly what you were talking about, how we use the tools of cognitive neuroscience to make progress, where the sort of challenges are, what we've learned really over the last, several decades of doing, human neuro. But let's get into it. I'm going to start actually going back to some of the early work. And so this is something which I think is familiar for most of the people in the room. This is the original characterization of the dissociation, the double dissociation between the what and where pathways in the macaque. And it's a really elegant piece of behavioral work replicated many times since, basically showing that when you lesion the posterior parietal cortex, you find a deficit in monkey behavior, which is critically dependent on spatial features, in this case, the adjacency of an object to one of two food wells. Whereas when you lesion the temporal cortex, you observe a deficit when the location of the food is given by an object pattern, right, where they have to form some long-term association between the structure of a physical object and then the right answer. And then with these types of damage, you don't observe deficits in the other tasks. And this leads to the characterization, right, of primate vision as being a bifurcation, where you have early visual input coming into the posterior, primary visual cortex, and then a split, right, with spatial functions ending up in the where pathway, and then non-spatial, visual detail ending up in the what pathway. This is an influential idea. It has absolutely been cited many thousands of times in many various forms. It remains quite fundamental to a lot of the work that we do today, particularly in our characterization of visual processing in the macaque and in the human as well. It is also an instantiation in particular of hierarchical processing, right? In that what it's really describing is a series of processing stages going from early vision through a sequence of areas in which simple representations are slowly transformed into more complex representations through a process of aggregation. And you see a fair amount of evidence that this is true, right? Where if you look at the latency of the so-called first spiking activity, you can observe the increasing delay. You look at the size of receptive fields, which is given by these circles, and they are increasing. And you also observe selectivity for increasingly complex stimuli as you work your way down the length of the temporal lobe, culminating in representations of individual highly familiar or personally meaningful things. If you instantiate this general idea of hierarchical processing into a computational model, you essentially get to the structure of the modern DNNs, right? Where what you've got is a series of layers that are going to be providing input in a stage-like fashion, and you have a bunch of non-linear aggregation mechanisms that are going to build towards more and more complex representations. And this has proved to be really computationally quite a powerful principle, but it does run into a bit of an issue. Which is when we started to do the human work and take it really seriously, one of the things that we observed very strongly, and Russell, you contributed yourself very strongly to this as well, and I will put in a plug and say your work has also been extremely influential in all the stuff which I do. What you observe is a stereotypical pattern of category selective areas. Right. Specifically, you find in general a paired structure where on the lateral and then ventral surface of the temporal lobe, you get areas in the brain in roughly the same location across people that respond most strongly to one category of visual object over some other category of visual object. And this, as an observation, played really well with, in essence, cognitive science ideas for how high-level visual processing was supposed to work, because you do very different things with faces and body parts and scenes, right, then across those different categories. So the notion of a specialized, localized representation that was going to support those specialized kinds of behaviors made a fair amount of sense. It also plays relatively well with the neuropsych, where you can find patients who would show at least some sort of preferential deficits for some of these categories as well. The issue is there's not really a good way of anticipating this structure from the general principle of hierarchical processing, right? Those models will produce complex representations, but there's no fundamental constraint within them that creates stereotypical locations or even necessarily clustering. This leads to proposals that maybe what we need to do in the human, and this is from a paper from Edward DeHaan and Alan Cowie that came out, coincident with the review papers I put out that Russell mentioned, where they propose that we should throw away the entire idea of the pathways themselves and instead start to think about these as a series of independently, in the strongest formulation of the theory, evolved modules, which were densely interconnected within particular categories, but only sparsely connected across categories, and then sort of located in an arbitrary way. This idea really amounts to basically a description of the data, and it comes with a couple of problems right off the bat. Right, the first thing is that it is arbitrary, as I said. There is no necessary relationship, right, between that observed structure and the structure that you observe in any other animal model, or even necessarily that you're going to observe for now new categories of things. So it breaks the relationship with the primate literature entirely, and then it also opens up a quest that will never end for areas selective for every given category that you can actually think of. And this deeply relates to the kinds of more computational phrasings of this problem, like dimensionality reduction, or trying to find, minimal sets of description. This essentially eschews that and says, we're just going to have to describe all of this forever. The ultimate solution, right, that actually works, I think, is to acknowledge one fundamental thing, which is that our models, our computational models of these hierarchical principles are just that, they are models. And they don't really capture the exact structure of the brain, which in fact has a great deal more detailed interconnectivity than these models typically appreciate. And so the argument I'm going to make is that we can actually get these things to play well together if we start to think about this a little bit more deeply. I think actually there are a couple of critical pieces of evidence. One is the mere existence of the visual word form area. And that is because this is an area which is selective for a particular visual category. There is, again, a deficit associated with it, alexia, where you find a preferential deficit for that visual category with damage in and around the area. It's stereotypically located, but there's no way to propose that this is the result of evolutionary pressure. Right? It simply can't be. Even being really generous in the American populace, general literacy is perhaps 120, 150 years old. And there's just not enough time for evolution to have placed a module here. And so what we need is a mechanism by which we can get the same stereotypical organization that we get out of category selectivity, but without proposing sort of special evolutionary events. And I think the answer to that is to be found in the details of the neuroanatomy. And so throughout the talk, I'm going to be citing particular work which I've published, but you'll see sort of images of people appear, and these are the people that were really critical in doing all the work. Here, I really want to call out Salim Khattar-Bacha, whose knowledge of the macaque neuroanatomy is simply encyclopedic. What he did was to look over the past 40 years of neuroanatomical tracer work that had been done since the original characterization of the dorsal and ventral pathways by Mort Mishkin and Leslie Ungerleider, who you see there at the bottom, and to really catalog and draw out a neuroanatomical skeleton. That skeleton is what you see here. Don't try to take it all on board. It's rather more complicated than it actually, I think you can get out of a single figure. But what it shows is that it is not the case that the brain is sort of homogeneously interconnected. There are particular connections and therefore particular areas that have more ready availability to information than some other areas might. As well, we can see that, the ventral visual pathway, for example, is not just sort of undifferentiated circuit that provides an abstract representation of category, right? What it's actually doing is making a deep set of interconnections with a bunch of other specialized areas, doing things like representing long-term memory for patterns and structures and mental maps, representations of value, anticipations of reward, and then structures associated with working memory as well. And so the argument that I will make is that actually the reason why we get this sort of stereotypical structure, and even down to the level of the more fine-grained characterizations of representation, like what I'm about to show you, is because of the constraints of the neuroanatomy. The neuroanatomy essentially determines where information is going to be broadly available across individuals in particular brain areas. Experience then acts upon those connections in order to shape them to deal with the particular kinds of visual stimuli and associated behaviors you're trying to produce. And that is where this stereotypical pattern of selectivity is going to emerge. And that's why even when you look for this pattern of selectivity for evolutionarily recent categories, you can observe it and it remains stereotypical. Here to talk a little bit about some specific work showing that, and really this is my work, but I think there's a ton of work that I could have shown you here. We can look at the representations of scenes, and we'll see that it bears out some of the predictions of that account. The nice thing about scenes, of course, I mean, you have Russell in your department, so they're really complex, which is great. They have a number of different possible features and dimensions which you can represent coming from the same image. Right? When you look at an image like this, you can be interested in the objects or the category or the geometry. On top of that, they're also very different from one another, right? And even though they are very different from one another, you are still capable of very rapidly extracting information from them. So what we're going to do is now try to see if we can predict the nature of representation in a particular area of the brain on the basis of its neuroanatomy. Right, so here is the parahippocampal gyrus. This is going to be the macaque homologue of the parahippocampal place area, say, that Russell described in the human. What we see in the macaque is that it is deeply interconnected, in particular with the ventral portion of the V4 visual field map. That is going to be a lower field, upper field representation rather, but also a high bias towards the peripheral portions of that map in terms of the projections. We can see evidence supporting that when we look at the human neuroimaging data. Here I'm just putting up a particular result, sort of classic result from Rafi Malik from the early days of imaging, showing a peripheral field bias as we move up the medial surface of the inferior temporal lobe. If we look at the descending connectivity coming into the area, we see an interesting pathway here, very well described in rodent. We were amongst the first, I think, to really characterize it fully in the macaque. What you're looking at here is a dense set of interprojections coming from the posterior parietal cortex, ultimately making connections throughout the medial temporal lobe, but in particular, this TFO, TFTH set of regions, which is the homologue of the monkey homologue of the PPA. There's a bunch of evidence in functional connectivity. This is a summary figure, I think, from Margoulis's paper from 2014, showing a similar pathway in the functional connectivity data in the human. What we're going to do, though, is now make, therefore, the prediction that PPA is going to represent the spatial aspects of scenes. That's because it's getting a bunch of its input from peripheral parts of the visual field, and it's densely interconnected with the parietal cortex, and then a bunch of other, even more specialized structures that contribute to navigation. We want to show that, though, in an unbiased way. And this sort of design element question we're going to come back to a little bit later as well. What we want to do is design an experiment that's capable of showing us that that hypothesis is correct without necessarily resorting to a sort of categorical contrast, right, where we're essentially designing one of our possible answers into the experiment. So what we're going to do is try to design something that's as data-driven as we can manage. We're still going to have to make some choices because we obviously can't show every scene possible in existence, but we're going to show a large array of scenes, in this case 96, spread out evenly across a number of different both spatial and semantic categories. So you can see some of that structure here. Here we have a bunch of caves and a bunch of mountains, highways. We have splits between man-made and natural, between open versus closed boundaries. We also have splits in terms of the apparent depth which is available in the scene, where, for example, you have here a hallway that's relatively short and here a hallway that recedes into the far distance. We're going to show each one of these scenes equivalently with an orthogonal task, no bias in the design at all, and then simply allow the response of the PPA, the multivariate response of the PPA, to show us how similarly each one of these different pairs of scenes is actually treated. We can then take that response and plot it as a multidimensional scaling plot, where now the distance between any two pairs of scenes is reflective of the distance in that representational space from the fMRI. You can see that configuration here. It's a little bit easier to see if I make it symbolic. Here's the exact same configuration, now colored by man-made versus natural. You can see that there's a fair amount of intermixing there, not a great deal of structure. Here is it coded for that apparent depth, near versus far. You can see a bit of a bias here for this spatial dimension, with maybe more of the near scenes clustering towards the bottom than the top. And then you can see a very clear separation for this effect of the overall extent of the spatial boundary, right? Whether or not that scene is open or closed. Again, this is also a result that has been replicated now several times with several different designs. But what we see here is a preferential sensitivity to the spatial features of scenes, which is exactly what we were expecting. Specificity is always a good thing to show. Here's the exact same analysis for the same scenes and the same subjects in early visual cortex. You see none of the same structure. We also want to close this loop. So a big theme in all of my research has been trying to make not just an observation within the physiology, but then a prediction from the physiology that leads back into the behavior in order to close that loop, with the eye eventually towards modifying our basic understandings of what the behavior actually is and what the mechanisms are that are generating it. So what we're going to do here is simply take the degree to which a scene is open or closed from the fMRI data. Roughly speaking, that's sort of how far to the left or right it is within that MDS plot. And then in an entirely independent group of subjects, we're going to arrange a set of matches between pairs of scenes where they simply have to pick the one that is more open or more closed. We then use the same math they use to rate chess experts, and that's what you see on the x-axis. So 1600 is a grandmaster in chess. It is also the most open scene in the set. And you can see the strong relationship between these two things, despite the fact that these are different subjects and different tasks. And so the only thing holding this relationship together is the stimuli themselves. What we can also see is, again, specificity. We do the same analysis with an early visual cortex, and there's no relationship at all. This tells us that the spatial aspects of scenes are absolutely critical. It comes right along with our predictions coming from the neuroanatomy. And so that's a good prediction. It shows that a good verification rather of a prediction. It shows that the structure, even down to a fine-grained level of representation, can be well anticipated by the neuroanatomy. The other part of that account I told you, though, was that experience was ultimately going to drive the formation of these representations. And so now we're going to do a completely different pivot into something a little bit newer. In order to investigate what the affective experience is and how much of an effect it has and how rapidly, we pivot to a large behavioral data set. So what I'm going to tell you about now is a study that we did in a data set gleaned from a smartphone game, which for some reason over 15 million people have now played. In this game, you take on the role of an airport security screener, and you repeatedly search bags for dangerous items. The makers of this game, as they were building it, unbeknownst to them, designed what is in essence a psychology experiment, in that all of the bags are generated randomly and procedurally. There is no actual statistical structure within this whatsoever. And every bag either contains a target or it doesn't. Because over 15 million people have played this, they have generated around now 4 billion trials worth of visual search data. And because 15 million people have played it, I have 15 million trial ones and 15 million trial twos. And that's going to allow us to look at the affective experience with a level of fidelity that frankly has not been possible before. So this is work that's really critically done by Michelle Kramer, a former graduate student in the lab, who's now actually working with the TSA on their research team, and Patrick Cox, who is a postdoc in the lab, currently on the job market. What I'm showing you here is essentially the results of a very simple analysis. On the x-axis, we have trial number from trial 1 to 10. On the y-axis, we're going to have accuracy for detecting a prohibited item, right, within a particular bag. What you're going to do is then titrate the performance as a function of the number of prior trials in which a prohibited item was actually seen. Okay, and what you can see already in the structure here is that the more prior trials include prohibited items, the more accurate you are or at saying that an item was actually prohibited. And the further into the trial sequence you are, the better your behavior overall actually is. Now, an eagle-eyed person would say, well, this is a complicated way of plotting this. We know how to aggregate these two things together. And indeed we do. We can take the amount of evidence and the strength of that evidence in a particular direction and aggregate it in the simplest, frequentist way as a binomial z-score with the following equation. We can then take every single data point within that particular plot and now transfer it over onto a single axis for evidence. And you can see that the prediction is remarkably strong with an R-squared of over 0.995. Nor are the effects small, right? Overall accuracy is going to vary by 15% from around 97% down to around 82, depending on the exact sequence of trials that came before it. This is actually data from a simpler task where all they're doing is saying whether or not a single item on the screen is a dangerous item, a prohibited item or not. We're also going to look at that within the visual search, which is a little bit more complex. Here, though, again, it's procedurally generated, and so they are searching for this target, and either the target is present or not. We can simply look for now evidence of target presence. But because this is a more complicated display, it allows us to separate out that effect from the effect of sort of aggregating evidence towards target presence or absence from the exact response that they're actually making or the presence of particular distractors. And that's going to let us separate out the different impact of motor response, cognitive set, and perceptual processing. First off, we're looking at evidence for target presence. We see exactly the same relationship here. This is a set of 1 million people. We actually, in this paper, internally replicate this 8 times on 8 separate sets of 1 million people. You get the same result every single time. What this shows is that every single trial that the individuals are experiencing shapes the behavior on the subsequent trials. Okay, and it does so via a remarkably simple metric of statistical evidence. There it is for hit rate. Here it is now for hit time. This is the amount of time it takes to detect the target, opposite going relationship with the same basic structure, which is exactly what you would expect. You're faster, the more evidence there is for target presence. What we can do now is still say, well, target absence or presence is defining the response on any particular trial, right? Your job is to find these dangerous items or not. So now we can look at factors which are orthogonal to the overall response. And so, for example, take a given distractor, right? So here we have the headphones, and you can have a pair of headphones in a bag that contains a target and also a bag which does not. And you can also be lacking a pair of headphones in a bag that contains a target or in a bag that doesn't. And now we can simply say, well, even though this is not directly driving response and is in fact orthogonal to it, is there still an advantage, right, for having experienced headphones in previous trials? And the answer is yes. Here are exactly the same relationships in now your ability to detect a target in the presence of headphones as a distractor is better the more exposure you've had to headphones as represented by the Z-score, and your time to detect that target is also faster. Okay, what this essentially tells us is that your experience, even with things that would be traditionally labeled as task irrelevant in a visual search task, is on every single trial causing a shaping of your behavior in subsequent trials. Okay, that gives us a profoundly powerful mechanism for shaping the structure of representation over the long term. Right, as you are dealing with incoming perceptual information like faces and scenes and body parts and whatever else it is, if those individual things are consistently both being processed and then generating particular kinds of behavior, right, you have particular types of processing demands. Therefore, with those categories, you can end up shaping representation quite profoundly and quite quickly. And that will eventually be reflected in the gross structure, the gross functional organization of the ventral visual pathway. And so it really requires us to acknowledge that our DNN models are models of a computational principle, right? They are not ultimately models of the structure of the brain. And as a result, they're not really going to give us a great deal of insight into aspects of things like functional organization. We can still learn a great deal about what hierarchical processing is capable of accomplishing, but I think we will also discover some of its limitations, and our ability to match it to what's going on in the brain is always going to be a bit suspect. Ultimately, we will need to understand a great deal more about the neuroanatomy, particularly neuroanatomy in the human, if we want to start to constrain our understanding and make tighter predictions of exactly the nature of representation that we're going to observe. And so what I've shown you so far is really the long-term shaping of representations in a way that's consistent with sort of ongoing experience and a mechanism, right, whereby even individual trial exposures appear to shape ongoing behavior, and concomitantly they're going to affect the structure of the physiological response. What I'm going to show you now is that these effects are not limited to just so-called perceptual or bottom-up recognition kinds of tasks, that even high-level cognitive processes are going to have an opportunity to cause the same sort of shaping. Okay, Russell mentioned some work in working memory. We'll do a sort of whirlwind tour of that. And I think we're good on time, so we might get to some even unexpectedly new stuff at the end of this. We were here particularly interested in what the nature of representation was for items that were being held in memory. And this is a particularly, I think, elegant study, which was done, which I did in collaboration with Soo-Yeon Lee. You see at the bottom here, now faculty in Korea, where what we did was to adapt Frank Tong's basic experimental design, where you see 2 different items, and then you are cued to hold one of them in memory during a delay. But we added a twist, which allowed us to probe the nature of representation a little bit more deeply. So here, instead of seeing 2 basic Gabor patches, what you're going to do is see 2 different objects. And then in different task runs, we asked participants to do a different task following the delay. Either they were doing a visual task where they were shown a sliver of an object and they had to say whether or not that sliver of the object matched the item that they were holding in mind. And in this case, it does match this little bit of the clock, so the answer would be yes. Or in the non-visual task, they were simply asked to say whether the category of object that they were presented with matched the item that they were holding in mind. And so here they were holding in mind a clock, which is not the same as a watch. And so the correct answer is no. But critically, in these two different kinds of runs, the actual stimulus train through the end of the delay is identical. The only thing which differs is the task demand that's going to follow that delay. We then run a standard localizer looking for object selective areas in the posterior cortex. And then this is a one-back localizer in order to find areas in the prefrontal cortex associated with working memory. What we then do is a decoding style analysis where we look to decode which one of these 6 items is being held in memory. That gives us our chance line there. And when you're holding a visual information in memory, right, we see significant decoding in PFS, but not when you're doing the non-visual task, even though you are seeing the exact same stimulus trains in the same subjects. But when you flip the task demand so that now it is a non-visual task, you no longer see the decoding in PFS, but now you see it in the lateral prefrontal cortex. That tells us that the nature of representation, right, is what is determining the recruitment of particular areas for holding information in working memory. If your task requires visual detail, then you will engage visual areas. If your task does not, then you can engage areas that maintain, for example, verbal labels. That tells us that working memory, right, may very well crucially involve even so-called sort of perceptual or visual cortices. Sorry, a little bit of reverb there. We want to, of course, make a behavioral prediction, which comes off of that, which I did here in collaboration with my former graduate student, Shunwei Ting, who's now in Brad Possel's lab, and I think also on the job market, actually. Oh, sorry, Russell. Yes, I will take a question.
Sorry, I've been trying to ask a question. It's just a simple question, which is I just missed what the non-visual task was.
Oh, so the non-visual task here is simply to say whether the category, right, of object presented as the probe matched the category of the object that they were asked to hold in mind. So here they're asked to hold in mind basically a clock, and then they simply have to say, is this a clock? And because it is not, then the answer is no. If it were a clock, we would have shown a different clock, and that would have produced the answer yes. Did that get to it, Russell?
Yeah, thanks. Just something I missed.
All right. Okay. So now we want to make a behavioral prediction, right? If visual working memory is actually directly recruiting these sort of perceptual areas, then there should be some sort of a perceptual interference effect, right, that comes along with that. And so that's exactly what Shunwei got interested in and then set out to prove in what I think is a really elegant set of studies, but they're also quite involved, so I apologize ahead of time, but I also will not take you on this journey unless it has a happy ending, so be assured that it does all work out. In order to show this, we're going to do a rather involved psychophysical study, conducted actually entirely on mechanical Turk. What we're going to do is first present a Gabor that has both a color and an orientation. And participants are going to be instructed to either hold in mind its color or its orientation, but not both. Following that, they are going to then do a discrimination task, where they are going to be asked to tell whether or not 2 colored Gabors are the same or different. Again, they both have color and orientation, but the discrimination is only going to be taking place on the basis of one of those 2 features. Okay? And afterwards, they're going to do a continuous report for the appropriate dimension, right? Reporting either the color of the Gabor or its orientation. Now, what we're doing then is a dual task, looking for the influence of the item that's held in memory, but now long since removed from the screen, on this discrimination task, which is the best behavioral task that we have for tapping into perceptual processing. And so we're going to manipulate a couple of things in order to show this difference. The first is, of course, we're going to manipulate the difference between the discrimination stimuli so that we can derive psychometric functions and then perceptual thresholds. And so we're going to have varying gaps in terms of the number of degrees in either the color space or the orientation space. And then we're going to ultimately derive these psychometric functions From those functions, we'll take the point at which people are 66% correct, and that's going to be their threshold for how big of a difference we say they need in order to consistently report the two items as being different. Okay, we're then going to manipulate our task instructions so that for different groups of subjects, we either end up with a match between the feature that they are doing the memory and discrimination on, or a non-match. For example, they can be doing color memory and color discrimination, orientation memory and orientation discrimination, or the crossings of those two. That's going to allow us to see whether or not our effects are specific. After all, there could be passive carryover from this presentation into the discrimination, and we want to rule that out and only observe the interference effects during the conditions requiring an active maintenance. The only difference, though, in stimulus trains between these 4 different experiments is the task instruction. Okay, and the final thing that we're going to manipulate is the relationship between the object that's being held in memory and the discrimination stimuli. Okay, on the following theory. Through a set of earlier experiments that I'm skipping over, but also intuitively from the finding that you do seem to see this activation, even pre-activation for particular populations when you're holding visual detail in memory, we can start to hypothesize about what sort of interference effects we should actually observe on these discrimination stimuli. So here we have the color spectrum. And let's imagine that these 2 dark sticks are the actual physical locations in the color spectrum of the discrimination stimuli. All right, now you're holding in mind a particular color. And we know that that is going to involve now, let's say, an increase in the tonic level of activation within a population of cells that are selective for that particular level of the feature, and then a fall off, right, with increasing dissimilarity from that point. Okay, therefore, we've pre-activated that population. Now we're going to present these 2 stimuli, and because we're using an overlapping set of cells in order to do the representation of the thing held in working memory and the information coming in via perception, we're going to predict that these 2 things are going to drift, right, in the direction of the thing being held in memory. If the item you're holding in memory lies directly between the 2 discrimination stimuli, they drift equally, because they're equally dissimilar from it, towards it. As they do, they move towards each other. And that means that the actual distance between these is going to be larger than the perceived distance, which in turn should mean a larger threshold needed in order to consistently say the discrimination stimuli are different. All right, now for comparison, if you're holding an item in mind that is off to one side of these 2 stimuli, they both still drift towards it, but the level of the amount of drift is determined by the overlap in the population, which you can see is different. So this stimulus drifts more than this one. That in turn means that the actual distance between them is smaller than the perceived distance, because they seem to be more dissimilar, and that should lead to lower thresholds. Okay, and we should observe that effect again only in our match conditions. Okay, to whiz through this, here is the raw data for now color memory and color discrimination in the middle condition. Here's that same experiment now for the side condition. Here are the psychometric functions. Here are the derived thresholds from there, and you see the offset exactly as you would expect, with a lower threshold for side than for middle. Okay, here's the exact same discrimination now for color, but when they're holding orientation in memory with the same stimulus train, and you see no significant difference between them. Okay, what we can do is then take those subtractions between the 2 thresholds. That's what's plotted on the y-axis. And now we're simply going to plot the difference in those thresholds between side and middle as a function of whether which feature you're doing memory on and which feature you're doing the discrimination on. And you can see that for when the conditions match, right, for color discrimination and color memory and orientation discrimination and orientation memory, you have a much larger interference effects than when they do not match, to the point of essentially extinction in the color condition. That crossover interaction then gives us direct and highly consistent evidence that there is this ongoing behavioral interference effect. We are right now conducting the studies, and I'll present a little bit of it, using transcranial magnetic stimulation in empty cortex in an analogous paradigm to show that when the area is stimulated, you also observe concomitant differences in both the veracity of the working memory report and a diminishment of the interference effects during working memory. With those results in hand, that's really strong evidence now that we have closed the loop and that these, so-called perceptual areas are deeply involved in working memory. But remember now that every single time you drive activity through a circuit, you are necessarily changing the structure of that circuit. And so working memory is now impinging on the structure of early visual cortex, early visual cortical areas, I should be more precise, and potentially changing its long-term structure as well. And that's another way in which the ongoing demands, right, not just of the behavior that you're producing or the processing of the individual stimuli, but the high-level cognitive processes that you're engaging also have an opportunity now to shape even putatively low-level circuits. Okay. What I'm going to do now is show you a little bit of even like very old work that I did in my graduate career, actually, which generalizes this case even a little bit further. And so here, what we're going to do is try to contrast sort of distributed processing models of how these systems function against the more traditional, almost box and arrow classical accounts for these high-level cognitive processes. And essentially the core difference, not to belabor it, is that the distributed view proposes an integrated circuit, which means that the consequences of changes in processing in one part of this circuit are necessarily going to spread to the other parts of the circuit as well. As I've just been telling you, that effect is very likely to then have long-term consequences for the structure of even distal circuits. What I'm going to show you here is a demonstration of this interactivity in the context of visual attention. Okay, the little inset there is Marlene Berman, who is my graduate advisor. I really do this set of studies because I never really get to talk about them, and they are kind of fun in the sheer number of different predictions that we are going to make. Okay, so one possibility is that attention, spatial attention, is essentially completely spatial, right? Which is to say, if I cue you to a location, then the speed with which you'll be able to detect a target at some other location is going to be a direct function of the distance between the cued location and where these other targets are. In this case, these near and far object targets, as labeled, are the exact same difference from this cue. But we also know that there is such a thing as object-based attention, where if I cue an object, I get a benefit for other locations within that object. In this study, what we did was to set out to show that that interaction not just benefits the object, but it also changes the structure of spatial attention. So if I cue a location within the object and the other locations benefit, they will also start to create gradients of spatial attention because they are being facilitated in the same way that I am facilitating via the cue directly, perhaps to a slightly weaker degree, but the same underlying process because you're driving activity into these circuits that represent those locations in space. As you do so, you pull the sort of group center of mass away from the location of the cue into the body of the object, and now the near object target is closer to the center of the object, right, than the far object target is, leading to the prediction that this should be detected more quickly than that. Okay, but we're going to make this worse because we're going to add a second object. So now imagine when I cue a location in space, I'm not just cueing that location, I'm not just cueing the object that underlies that location, but I'm cueing the abstract structure of that object as well. In this case, let's just call it rectangle. Okay, so if I'm cueing rectangle, this location, and the object, I pull the center of mass down because of this facilitation within the body of the cued object, but now there will also be some facilitation within the uncued object by virtue of sharing a structure. That in turn pulls the center of mass towards fixation, which is also equidistant from these two targets. Okay, contrast that with a case where we have a second object that does not match, in this case, its particular shape. There will be less of a facilitation of the locations within this object, and therefore less of a shift towards fixation and a recovery of the difference between these two targets. We also make the straight prediction somewhat simpler, that shifting between these two objects should be a little bit harder than shifting between these two objects, even though that spatial distance is exactly the same. Okay, long story short, all those predictions hold. They hold for shape. They hold also for color. Here you see the equivalent experiment where you see a larger cost to shifting between objects in the non-identical displays than in the identical, and then a greater difference between the near and far object targets in the non-identical displays than in the identical as well. This generalizes beyond, so-called low-level visual dimensions here is an equivalent experiment done with high-level category. You get exactly the same pattern of results. I particularly enjoy this one because it took me a day to figure this out, but the lowercase h and the 4 are the same object with an 180-degree rotation, which means that between these two displays, you have a very different pattern of behavior, even though the exact same pixels are actually on the screen. This is therefore a very clean demonstration of a high-level semantic effect within the context of a very well-controlled and, putatively low-level visual attention task. Okay, so what do we get if we put all this together, right? What we end up with is a system that is ultimately going to require a great deal of us as empirical scientists in order to successfully interrogate, right? A lot of the observations that we get out of the system are going to be ultimately related to neuroanatomy and experience in some combination. That's going to require us to have a deep understanding of neuroanatomy, which we currently, frankly, lack, particularly in the human. A deep understanding of plasticity, if we are to understand how we get from, the case of, quote unquote, novice brain in young children, and we get through the course of development to more adult structures. And it's also going to require us to understand experience, but at a level of detail and fidelity that we are really going to have to work hard in order to interrogate. Because it's not just that, okay, you've seen this many stimuli this many times, right? Ultimately, the effective experience is not going to be completely stimulus computable, precisely because these high-level processes are going to have impinged on the structure of even putatively low-level visual representation. And so to understand this system fully is really going to require a bunch of experimentation. It's going to require an enormous amount of us, an enormous amount of different methods. But frankly, it's the reason why I have always been excited about being in this field. I think this is perhaps the biggest challenge in science, right, is to try to measure a thing successfully and understand it, even as it is adapting its structure to the demands of the way that you're measuring it on a trial-by-trial basis. Okay, and so I want to leave it there to leave time for questions. And, there are also some, less well-developed things that we can discuss if there's going to be time left at the end. But in the meantime, thanks for having me, and I will take any questions that you might have. Thanks so much.
Is there a question in the chat?
There is a question just about somebody looking for the citations for the talk.
Oh, okay.
I think you can see that.
All right, great.
Yes.
So in the room. Yes.
Hi, yeah, that's great.
Can you hear the question?
Hmm?
Can you hear?
Yeah.
Can you hear me? Okay, cool. When you showed the multidimensional scaling results for the scenes and showed that you can differentiate between certain spatial features like indoor, outdoor, near, far, I also noticed that there were some consistent low-level visual feature differences between them. Like, for example, the open scenes also had a lot of sky and they were more blue.
Oh, absolutely.
These had sort of warm light. Do you think you can account for those things?
So, I'm glad you asked this question because I think it gets at a deep issue that we're going to have to grapple with. So you ask exactly the same question that a number of the reviewers ultimately asked. And our response to it was to do, I think, what everybody does, which is a bunch of analysis of the low-level visual features within the scenes to see whether or not things like differences in spatial frequency or, complexity or contrast or whatever it was explained any of the categorical differences cleanly. The answer there was no. What I'm going to do, though, is qualify that in a way that I think applies beyond the boundaries of this study a bit. I think absolutely there are going to have to be low-level feature differences that define any natural category. Right. So my one response, and I think it's the knee-jerk response that we all try to have because we want to try to dissociate our high-level things from low-level things, is no, this stuff can't explain it. But that's ultimately a ton of nulls. And, if I gave a more complicated answer, like clearly if I put a DNN on those images and I trained it to provide me the category label of open versus closed, it would succeed, even if it's completely a feed-forward architecture, right? I can train it to do that, which means that is some ultimately combination of features, right, that's going to ultimately drive any of these categorical differences. I think that that's ultimately a feature and not a bug, right? What we want is to describe what those sets are. And I think the fact that the representation emerges is no less interesting, right? Why is this particular constellation of features actually got a representation that appears to, apply over this much of the actual cortex, right? It's not like this is the only high-level nonlinear aggregation of low-level features that could have been represented here. So why is this the one that's actually there? And I think then we come back to the answer of, okay, here's the neuroanatomy that provides, perhaps preferential access to different subsets of those features. And here's then the task demands, which are ultimately driving the way that information has to be processed to produce adaptive behavior. But I think your question is an excellent one ultimately, because we want to really describe what the features of the image are that are contributing to our representation in the same way that we want to describe what the processing and task demands are that are contributing to that representation as well. To just round that out with a more specific answer, we did look at things like, amount of blue or particular color. It really doesn't do a great job, mostly at the level of the detail. So absolutely, like there's more sky in these scenes than there is in the closed scenes, but exactly which scenes end up as the most open and most closed was not particularly well predicted by that at all. And so, but I do think there is going to be some interesting combination of features that define this. I hope I got to the answer there. I can't see you, so I don't know.
Oh yeah, that was great. Thank you. I was nodding the whole time.
Okay, awesome, excellent.
I'd like to ask a sort of general question, building on what you said at the end, which is you said, oh, we know a lot about the macaque. We know less about humans. I mean, you have these beautiful diagrams in macaque with these nice labeled ovals for regions and the specific connections between them. So how are we going to get that for the humans? And I mean, what is really the granularity of which we should be, looking at cortical organization? I mean, like the PTA, right? As you know, or as you alluded to, is several regions potentially in the macaque. So how do I get to the ground truth so I'm not sort of just saying these things like parahippocampal place area, which are probably not the real underlying unit, and is that even a meaningful thing to get at?
No, I mean, you're asking really, I think, the deep fundamental question for which I do not have a complete answer. Absolutely not. I think, and I'll generalize this a little bit to a broader point, and then I'll try to provide an answer. So, I have, in my position now at the NSF, I see a bit more of the work that's coming through the microscopy, where they're starting to do like wide field electron microscopy. And it's interesting because they are coming to the exact same question, but from the opposite direction. So as they are seeing more and more of the system, they are asking, well, how do I know now what the units are by which I should be subdividing this? And they realize that a lot of the subdivisions that they are coming to by default are basically handed to them by the prior literature. So it's like, well, okay, here's a synapse, and here's, here's the matrix which is supporting the health of that synapse, and there's a mitochondria within that. But then, does this synapse relate to this other one? Should I think of them as a unit? How do I determine this? And these questions are just as thorny. I think we really do have a fundamental problem in knowing exactly what the unit of analysis is that we can reasonably get to. And an enormous amount of the answer to that question that we have come to has been essentially defined by method, right? So as our scanners have picked up greater and greater resolution, we have started to talk more and more about subdivisions. Does that necessarily mean that those subdivisions are more fundamental than the broader characterizations we had before in a ground truth sense? No, you just can see them now. Right? I think the ultimate utility of these things is going to be in, do they make an interesting novel set of predictions that are testable, right? Does the observed structure I get out of this metric now give me some additional insight into something else in this broader system, right? Be it behavior or data coming from a different physiological method, or targeting that allows me to show some causal difference between these different subunits. I think in the end, though, the answer may be somewhat degenerate, which is the more detail we look at this thing in, the more reliable detail there is, right? I think some of the calcium imaging results, in fact, suggest that. It's like, well, how much information is there in this population of cells? Basically, the number of cells that you measure, right? It's going to be incumbent upon us as theorists to start to articulate where we think the generalizable information is, to show that that information is generalizable, but also not to become dogmatic about the particular ways that we're cutting this system up, right? And I think ultimately we're going to want to do a great deal more work, which is quantitatively convergent, right? Give me a bunch of measurements, make sure that the raw data is available in some usable form, give me a theory that makes some interesting predictions, but make sure that I have the observations. So when the next person articulates a theory at a slightly different level, we still have the data to actually look at it and see whether it holds. Sorry, yeah. Go ahead.
I mean, what about something simple like cytoarchitecture? As you're like giving this answer, I was thinking about like the work the colony growth specter gave relating functional regions to cytoarchitectonic divisions. I mean, it seems like there's got to be some fundamental anatomical things, but you're suggesting that even that is really kind of in the eye of the beholder.
I think in the end it may well be. Like what we could do is use that as an alternative parcellation, right? So we could say, let's imagine I gave you a perfect map of the cytoarchitectonics. Now, I will say there we don't have nearly as much of a handle on the individual variability, and I'll also say that as pretty as these macaque pictures are, we don't really have a handle on variability across individuals there either. But let's imagine we did. It's relatively consistent. Okay, here's my cytoarchitectonic map. Now I give you your areal boundaries out of high field fMRI, and they're going to agree in some cases and they're going to disagree in others. Are you going to dismiss the bits that disagree in favor of the cytoarchitectonics, even if these are making interesting behavioral predictions when you look at the information available in those things as areas, right? Like it's a little bit, we're going to find mismatches, 100% we are. And I think the trick for us as a field is going to be to come to the best descriptions of how much of the variability our ideas are accounting for and how much of the variability they are not. I don't know that we can a priori settle on any one of these different metrics as being closer to ground truth, because I think all of them have complexities embedded within them, imperfections in the measurement. And beyond that, is it necessary for a functional unit to completely respect the cytoarchitectonic boundaries? No, right? Like in the end, there's a bunch of synapses, and clearly there are connections between the cytoarchitectonic areas anyway. So why can't that connection form a functional unit at the level of what you're trying to do, which is say describe scene processing or describe how a particular complex behavior is created, right? It's just going to be thorny. I don't think that we can fall back on any one measure or method or analytical approach even. I think we need quantitative convergence, standardization, and as much precision and accuracy as we can wring out of this system. And in the aggregate, hope that we discover, where we have generalization and where we don't. And I think both can be informative.
Anna Shapiro has a question on Zoom.
Okay. How do we do that?
Hello, can you hear me? I know that Dwight can hear me. Okay.
Yeah.
Hi, thank you so much for the great talk. I think I might have missed part of the argument you were making, so maybe you can just sort of summarize it again. But I was very interested in what constraints you thought the TSA experiment was putting on your theoretical framework. In particular, like, are you assuming that those learning effects that you're seeing are occurring in perceptual areas?
Oh no, I mean, I think those individual learning effects, right? That's not physiological data. I think as we begin to separate out the task relevant from task irrelevant stimuli, we start to interact with older ideas about how that processing is structured, right? So for example, within the attention literature, there's a ton of work talking about sort of pre versus post selection and making the argument that the task irrelevant features may not be the subject of the same kinds of learning effects and that you need attention. And I think it speaks to those underlying issues. I don't think on the basis of that work, I would put a strong bet down on exactly which circuit is involved. beyond that observation that even these sort of simple and rapidly dismissed features are having an effect. I think the general argument I'm making there, though, is wherever this effect is occurring, right, you are ultimately seeing a trial by trial aggregation of information, which is quite profound, right? It means that the behavior that you observe in your paradigms, at a first stage, is ultimately constrained by that paradigm, right? It is optimized to the experiment that you ran. And that's an important thing to keep in mind. But I think it also speaks to the extraordinary plasticity of this system. So I haven't shown it here, but this is a self-paced game, right? So you run that experiment. Well, people pick up the game and they play a level, and then they put it down and they pick it up whenever they want to, and then play it again. And so what we've done now, which is actually in prep, is we take the predictions from the first level of the game and then try to predict behavior on the second level as a function of time. And what you actually observe is that you get stable predictive effects over gaps of even days, right? You can come back days later and these learning effects are still there, right? So it's not just that you have some sort of, some instantaneous cognitive set that you're forming, or some slow calcium kinetics, which are priming particular synapses. You really do have a stable change. And so my real argument there is just to say, look, this system is extremely plastic. And there's a bunch
Which system?
Oh, the brain itself overall.
Okay, the entire brain, I see. Got it.
Yeah, I mean, I'm not putting down a marker there at all.
Got it.
I think there's a lot of other evidence I could pull in that suggests that we have these sorts of learning effects in specific systems, but I'm really just saying we need to think about that plasticity and how long-lasting even single events appear to be on the shaping of behavior and concomitantly whatever the neural substrate is that's producing it.
Cool. Yeah. Thanks so much.
Absolutely.
No more online.
Okay, is there another question from the room, or? Yeah,
I'm thinking about this plasticity and about its relationship to the different areas that you're trying to explain. And I wonder if you would imagine that this plasticity would be greater within a functional area than if you had a task that was between functional areas. So if your TSA task wasn't to find an object among objects, but something like, I don't know, plan a route over a gigantic face. So, you have to do something that unconventional, like do a scene-like thing with faces, or, like, would you expect that there's some kind of learning constraint on the plasticity that respects the functional area boundaries?
That's a really interesting question. I think on the one hand, for particular transformations that are anticipatable from prior experience, even if like that event isn't anticipatable, like you do typically, like if I gave you a navigation task in scenes, you do a lot of navigating in scenes. And so might you have built a system that is more capable of adapting itself to the particularities of the situation it finds itself in for that task? Absolutely. In which case I think your prediction would hold, right? If I find a very unusual task that requires a novel kind of set of interactions across, multiple areas, that might be harder to do. Where I am more hesitant is actually physiologically whether that constraint is going to hold or not. Like if I were to do that as opposed to doing it paradigmatically, but do it via direct stimulation, I would be less willing to take a firm guess, actually. But I think it's absolutely an interesting question, and exactly the kinds of question that we have to get at, because I think we also want to make predictions, right? How much plasticity do we get within across areas is important. Or do we pick up differential delays, right, in the optimization of behavior if we go to more temporally precise techniques as well, right? There should be some consequence of having to coordinate activity over a larger circuit than a smaller one, right? At a minimum, you're taking in, potentially more hops. So there should be temporal or energy use effects there as well. These are exactly the kinds of questions that we have to unpack. And I don't know of at least the right data to give you a firm answer yet. Was that helpful? I hope so.
Any other? All right, Dwight, thank you very much. That was super interesting as always, and sorry you couldn't come in person, but I guess the Zoom worked okay.
Yeah.
Certainly very great, and really enjoyed your talk. So let's thank Dwight.
Thank you guys so much for having me. And you know, I'm always available on email. So if you have any follow-up questions or anything, just go ahead and drop me a line.
Thank you.
Great.